Welcome back to All About RX. Today, we're diving straight into the incredible physiological anatomy and the exact molecular mechanics behind skeletal muscle contraction. It's a truly fascinating biological engine, so let's jump right in. To put things into perspective for you, skeletal muscle accounts for approximately 40% of your total body mass. That's a massive amount of tissue. These are the long, cylindrical, multinucleated, striated cells that are wired directly to your somatic nervous system. Every single time you walk, talk, or lift something, you are relying on this highly coordinated network. Here's our roadmap for today's explainer: anatomy of skeletal muscle, the molecular sarcomere, types of muscle fibers, the neuromuscular junction, sliding filament theory, and finally, a clinical focus on myasthenia gravis.
All right, Part 1. Anatomy of skeletal muscle, the macroarchitecture.
Let's peel back the layers of a whole muscle, literally. We can zoom hierarchically from the whole muscle down into bundles called fasciculi, and then right into an individual muscle fiber. Inside that one single fiber, you'll find hundreds to thousands of tiny thread-like structures called myofibrils. It's kind of like a complex biological cable, where smaller wires are packed inside one another for maximum strength.
Enclosing this entire individual muscle fiber is a thin membrane called the sarcolemma, which actually fuses right into your tendon fibers at the ends. Inside this membrane, you've got the sarcoplasm, which is packed with mitochondria pumping out ATP, and the sarcoplasmic reticulum, which, as we'll see soon, is absolutely crucial for storing and regulating calcium. Moving on to Part 2, the molecular sarcomere, the micromachinery.
Zooming in even tighter on those myofibrils, we hit the microscopic machinery of movement. The basic repeating contractile unit here is the sarcomere, which spans about 2 micrometers between successive Z-discs when relaxed. And the structural stars of the show? 2 key proteins. You have the thick filaments made of myosin, about 1,500 of them per myofibril, with tiny projecting cross bridges. Then you have the thin filaments made of actin, around 3,000 per myofibril. These are actually made of G-actin subunits, wrapped up in a regulatory ribbon called tropomyosin and studded with the troponin complex.
You know those classic stripes or striations you see on skeletal muscle? They come entirely from this precise overlapping arrangement of actin and myosin. We've got the I band containing only thin actin filaments, the dark A band where myosin and actin overlap, the H zone right in the center with only myosin, and the M line anchoring it all right down the middle.
Now, how does this massive molecular puzzle stay so perfectly aligned? Enter titin. It's a giant, springy scaffold protein anchored to the Z-disc. It keeps the myosin and actin perfectly spaced and gives the muscle its elastic recoil so it can snap back to its resting state.
Part 3. Types of muscle fibers, performance variations.
So this microscopic setup actually dictates our real-world physical performance. Skeletal muscle fibers are classified by their speed, metabolic profile, and fatigue resistance. You've got Type I, slow oxidative fibers, great for endurance and posture. Then Type IIa, intermediate fibers. And finally, Type IIb or IX, fast glycolytic fibers. These burn out fast, sure, but they are uniquely tailored for rapid, super powerful short-duration movements like heavyweight lifting.
Part 4. The neuromuscular junction, the spark. We have the machinery categorized, but how does the somatic nervous system actually spark the movement? It all goes down at the neuromuscular junction. A large myelinated motor axon terminal sits in a synaptic gutter right above the muscle membrane. They don't touch, though. There's a tiny 20 to 30 nanometer gap called the synaptic cleft. The muscle side is highly folded into subneural clefts, and the nerve side is loaded with vesicles full of the neurotransmitter acetylcholine.
Here's the rapid-fire sequence. When an action potential hits the terminal, voltage-gated calcium channels fly open. This calcium influx triggers exocytosis, dumping roughly 125 of those synaptic vesicles worth of acetylcholine right into the cleft. The acetylcholine binds to postsynaptic receptor channels on the motor end plate, opening them up for a massive influx of sodium ions. This creates an end plate potential, totally depolarizing the muscle membrane.
Part 5. Sliding filament theory, the engine.
Once that electrical wave sweeps along the sarcolemma and down the T-tubules, the sarcoplasmic reticulum unleashes its stored calcium into the sarcoplasm. Now it's go time. That calcium binds to troponin C, shifting the tropomyosin ribbon out of the way. Boom! The active myosin binding sites on the actin are finally exposed, and the sliding filament theory kicks into gear.
Powered by ATP hydrolysis, the high-energy myosin head latches onto the actin, forming a cross-bridge. It then fires off a power stroke, literally yanking the actin filament toward the center of the sarcomere. The crazy part? It only lets go when a brand new ATP molecule binds to it. As this rapid cycle repeats, the sarcomere shortens. But remember, the filaments themselves don't shrink. They just slide past each other, meaning the A band stays beautifully and perfectly constant in width.
But hey, a contraction isn't very useful if you can't turn it off, right? This whole process shuts down in mere milliseconds. An enzyme called acetylcholinesterase rapidly destroys the acetylcholine. Some diffuses away, and active pumps fiercely shove the calcium back into the sarcoplasmic reticulum. No calcium, no binding. The contraction halts instantly.
Part 6. Clinical focus. Myasthenia gravis. When the system fails. So what happens when this intricate biological ballet breaks down? Take myasthenia gravis, an autoimmune condition. Here, rogue antibodies mistakenly attack and block those postsynaptic acetylcholine receptors. So the nerve fires, the acetylcholine is released perfectly, but the receiver is blocked. This drastically cuts signal transmission, leading to progressive, debilitating muscle weakness and even paralysis, simply because the sliding filament theory never gets that calcium trigger it needs.
It really makes you think, doesn't it? If a single rogue antibody blocking a tiny receptor can dismantle this entire microscopic engine, what other invisible physiological wonders are we just taking for granted every single time we take a step, lift an arm, or even blink? If you found this explainer helpful, please give it a like, subscribe to AllAboutRx for more science explainers, and drop a comment below on what we should cover next. Catch you in the next explainer.